Hello friends, welcome to the class. Today I will try to be a little louder and I have ordered my mic, soon it will come. So meanwhile you can use your earphones, and today's topic is membrane transport. So what is membrane transport? It is a process by which molecules move from one side of the membrane to another side. It is the most important function of biological membrane by which chemical composition and processes are maintained for cell and organelles. So permeability is a property of biological membrane by which it permits molecular transport, and all biological membrane are selective permeable, right? Means they permit transport of some molecules but not the others. So it has been divided in vesicular transport and transport across membrane. What is vesicular transport? In older literature of cell biology, membrane transport, uh, it is a classification on the basis of older literature. This vesicular membrane transport applies to macromolecules where the vesicles provide an envelope to mediate intake or secretion of substance. So like this endocytosis, it with the help of a vesicle it is taken inside the cell, and with the help of vesicle it is excreted outside. The process is exocytosis. This is transport across membrane. It applies to ions and small molecules. These substrates cross the lipid bilayer, this is a lipid bilayer membrane, either directly or through with the help of transport protein. We are going to study in detail about the transport across membrane. So there are two types of transport across membrane. One is passive transport, another is active transport. Passive transport, external input of energy is not required. System has sufficient free energy in the form of chemical gradient, charge gradient, electrochemical gradient. So this is the diagram. It is a lipid bilayer, and we have a concentration gradient which will allow to move from higher concentration to lower concentration, and if with the help of a transport protein the movement takes place, then it is the facilitated kind of transport. We will see the passive transport in details. Now the active transport, external energy input is needed, provided by ATP hydrolysis or protein motive force or light energy input. Input, this is for input. Input is necessary in order to overcome the adverse concentration gradient, charge repulsion and size barrier. So if in the flowchart diagram we will see membrane transport, it is divided in passive. In passive we have simple diffusion, for example oxygen, CO2 in respiratory. Here we have a direct passage of substrate through the bilayer. Then we have facilitated diffusion, chlorine ion and calcium ion. This is with the help of a protein, for example glucose uptake. And in active transport we have primary active transport and secondary active transport. In primary active transport, direct energy coupling between input and desired transport protein takes place, like sodium potassium pump. And in secondary active transport, indirect coupling of energy and intent transport takes place. So now we will see in the detail about the passive membrane transport. Simple diffusion. Substrate crosses the lipid bilayer without any transport protein. Substrate moves along its concentration gradient. Simplest and energetically most economical process. The substrate must be nonpolar, uncharged, small, and preferably lipophilic. This is used by respiratory gaseous exchange and lipophilic hormones such as steroid and thyroid hormones. The diagram is, it is a lipid bilayer structure. Due to concentration gradient, it will move from extracellular space to the intracellular space. So this is simple diffusion. Now this is facilitated diffusion. It is also known as aided diffusion. Requires a transport protein. This is a transport protein, and the flux rates may be higher and may show greater substrate specificity. So the diagram is like this. This is a protein, transport protein in conformation 1, where it is open from outside and closed from inside. In the T2 structure, it is closed from outside and open from inside. So the glucose will bind to the transport protein, and it will cause a change in the structure of a transport protein, which will allow the intake of glucose from outside to inside. So the transport protein binds to the substrate, go through conformational change and move the substrate on the other side of the membrane. For example, glucose transport in mammals. Okay, this was about the passive transport. I would like to give you a homework. Just in the comment section, write a short notes on aquaporins. Are they a active transport or a passive transport, and how does it function and where they are present? Now the active transport. There are two types, primary and secondary. Primary is the most common active transport. Direct coupling between energy input and desired transport membrane takes place, and for example sodium potassium pump. We will see in the detail in the next page. Now the secondary active transport. In this process, energy coupled is indirect. The energy input is not for directly driving the desired transport, but it is for creating a condition for the movement, like sodium dependent glucose transport. This is a diagram which depicts the primary active transport. This is a transport protein. In the initial stage, it is closed from outside and open from inside. 3 sodium is taken from inside. It will bind to the transport protein. It will cause an ATP phosphorylation. A phosphate group will bind to the alpha subunit of the protein, which will leads to the change in the conformation of the protein, so that the 3 sodium will get outside. It will expelled outside. The pump will open from the outside, and 2 potassium will be accepted from the outside. So the 3 sodium is getting out and 2 potassium is getting inside in this process. So 2 potassium will bind to the transport protein, and with this binding, dephosphorylation will trigger the conformation change. It will come to the initial position, and 2 potassium will pumped inside the cell. So this is an example of sodium potassium pump, which is an active transport. This coupling is directly bind to the desired transport protein, right? This is an diagram for secondary active transport protein. Here, a 1 sodium is going to bind to the transport protein. When it will be bound to the transport protein, it will cause a conformational change, which will leads to the binding of a glucose, right? The glucose will bind to the transport protein and pump opens to inside. Meanwhile, there is a process of sodium potassium pump, which will leads to the expulsion of sodium from inside the cell to outside, which will cause glucose to come inside the cell, right? So as the glucose is released inside the cell, the transport protein will come into its initial state. So this is a sodium dependent glucose transport. That's it for the today's membrane transport chapter, and today only we will do endoplasmic reticulum, so it is advisable to read at least once before you see the video. Thank you.